Welcome to Answers from the Lab, a podcast from Mayo Clinic Laboratories, where we explore recent trends, dive deep into emerging innovations, and unpack developments shaping clinical diagnostics. Hello, I'm Dr. Bobbi Pritt, a clinical microbiologist and laboratory leader at Mayo Clinic, and your host for today's episode. I'm especially excited for this episode as it's our first one in a new format we've been working on this summer. First, Mayo Clinic Laboratories CEO and President, Dr. Bill Morice, will join us to discuss some recent news. And then we'll do a deep dive on therapeutics with Dr. Paul Jannetto, our Vice Chair for Clinical Pathology. Let's start by visiting what's new and noteworthy in clinical diagnostics. Bill, welcome back.
Yeah, it's good to be back.
As always, it's so great to have you. You know, we have this new podcast format that we're really excited about, and we'll get to see that for our very first podcast here in this new format. And then we have a great topic lined up. So we really enjoy sharing timely topics with all of our listeners, but we also like to dive deeper into some of the most prominent industry advancements in clinical diagnostics. And this new discussion format allows us to do both.
Yeah, no, I'm very excited about it. It's as well. I mean, it's a natural evolution for, what really took off during the pandemic, and now giving it like kind of a little bit of a more organized format, so we can kind of cover the waterfront as well as dive into specific issues, or you can as well with other guests, and so we get more perspective shared here. So it'll be great.
Yeah, absolutely. Well, for our listeners, we hope these deep dives will help you understand how emerging trends may impact your lab and help you be a thought leader on these topics within your own organizations and communities. And for those of you listening, we would love your feedback on this new format. So if you have a topic you'd like us to explore, email it to mayocliniclabs@mayo.edu. So, for our topic today, Bill, I was thinking drug resistant infections is something we really should talk about. The World Health Organization just released an alert that drug resistant, what we call superbugs, are spreading. And in my world, the microbiology world, this is something we've been talking about for decades, but it's really getting to that point now where I think the whole world needs to pay attention.
I completely agree, and it's interesting, particularly for you and I, who travel different parts of the globe and really think about this, and parts of the globe where antibiotic use in stewardship is not as advanced, maybe, or even parts of this country or there and then health systems. So in my mind, it's like if you look back at the medical history, it was infectious disease as the scourge of mankind and the killer, and then as we
Right.
had the introduction of antibiotics, it pivoted to cancer, metabolic disease, and exposures, smoking, and other things. Right. The topic of antimicrobial resistance, actually, you could see a reemergence, unfortunately, of infectious disease and multi-drug resistant pathogens as like the new scourge. So I think it's
Absolutely.
very important that we talk about it.
It was really the 1930s, 1940s. We had antibiotics with penicillin developed by Alexander Fleming and others. 1928 started being used in the 1940s. Before that, we had sulfonamines, salvarsan. That completely changed clinical medicine. And as you mentioned, people used to die of infectious causes. Those were the top 10 reasons of death. Now it's malignancy and heart disease. But we risk losing all of that. And the World Health Organization, WHO, compiled data from over 100 countries, really cautioning that increasing resistance to essential antibiotics are now being seen. They found that 1 in 6 infections worldwide was resistant to the most commonly available antibiotics. So we are getting to a point where there are some infections we cannot treat at all with any of our existing antibiotics. And the WHO report shared that between 2018 and 2023, antibiotic resistance rose in over 40% of the pathogen antibiotic combinations monitored. And that's with an average annual increase of 5 to 15%. So scary stuff.
Yeah, for sure. Wow, those numbers are daunting, or concerning, even more than daunting. And there really is, on the one hand, you can say, well, gosh, is it really that big a deal? There's always new antibiotics coming out, and et cetera, et cetera. Well, it is, the worst end state is you have something which is not treatable, right? And then, then you really, but it's not treatable. So I
Mhm.
think that's enough said right there. But even if it is treatable with newer antibiotics, it drives up the expense, right? It drives up the use of those antibiotics and then the emergence of resistance. And that's when you get to the untreatable infections, right? So there's, and the convenience for patients, access, a lot of the more advanced antibiotics are not available in other parts of the globe. And the biopharma industry as well. I don't know that antimicrobials are a major focus of investment, right? They're more focused on personalized medicines, cancer vaccines, some of those things. So
Yeah.
because these are not the blockbusters oftentimes.
Right. There's a few things here. One, as you said, the new antimicrobials that we have are incredibly expensive because it takes a lot of investment, a lot of dollars to produce these new antibiotics. Second, they're not widely available. Third, there's actually not enough coming out in the pipeline, despite the fact that we do have some exciting new antibiotics on the horizon. There aren't enough of them. They are not coming out fast enough to overcome the resistance that we're seeing. And then, fourth, I'll just mention that, as you mentioned, this is not the focus of the pharmaceuticals. They pretty much lose money on these antibiotics, and most of the time, an infection is treated for a short period of time. It's not treated indefinitely. Whereas if you have heart disease or high blood pressure, you could take a pill every day for the rest of your life. So there's less financial incentive there. And it's hard to recoup the costs, which can be quite daunting for discovery of these new agents.
Yeah, well, maybe that's an area where AI can, because AI is really changing the game in drug development. So maybe that holds out some hope here, with
Yeah.
these things, because and the nice thing with them, with the microbial pathogens is you can really study their enzymatic pathways. I think are quite well studied. You know that better than I,
Mhm.
but there's certainly a connectedness to global health and the global use of antimicrobials that has a real impact. And so, it's if anyone likes to go to the gym, tinea corporis is something that you can face, right off of equipment. And now there was a report that due to the widespread use of like terbinafine as an oral agent in parts of the world, patients are undertreated. These are prevalent. We're now seeing terbinafine resistant, tinea. And that's I mean, yeah, it's not life threatening, but I mean, it really is something that could be quite unsettling. And again, the second line therapies are the ones that are used for very serious systemic fungal infections. And will we now see the emergence of resistance to those if these drugs are wide use? So it's again, it really is. It's like it is an ecosystem and an ecosystem problem when we put antimicrobials
Mhm.
into the environment, into the microbial ecosystem, either for humans or also for
Animals.
livestock, animals,
Mhm.
right? That it has an effect that it can boomerang back to all of our individual health. So I guess my
Absolutely.
question, and probably if that's my question, I bet it's a lot of people listening to this question is what do we do? What are some of the things we should be thinking about?
Yeah, this is such a great topic because we think of antibiotics, antimicrobials, and we think, well, that's the prescriber and people should just stop over prescribing. But we have a real role to play in the laboratory. It's very important. Diagnostics. We need diagnostics that are fast and accurate so that we can help our patient facing colleagues prescribe targeted therapies. They need to be accurate so that they're trusted, and they need to be fast so that they're actually clinically relevant. We've done studies at Mayo Clinic showing that we can perform a rapid diagnostic that identifies genes associated with resistance. But if we don't have someone to give that information to as soon as it's available, and that person's not going to be able to act on it, then you lose any benefit of having a rapid diagnostic. So we're the essential part of the system where we provide a diagnostic that's accurate, trustworthy, shows the resistance genes that are clinically relevant, but then we have to develop that partnership with the person who's receiving that information, whether it's a pharmacist or the provider. And then that person needs to act upon that information. So a diagnostic could be helpful in saying that something is a virus versus a bacterium that's infecting the patient. If it's a viral infection, let's say a respiratory virus infection, well, then that person doesn't need an antibiotic. That is not going to be helpful for that individual if you are giving them an antibacterial agent. So, not just detecting resistance genes, but just sometimes giving basic information about the type of pathogen a person has is so critical.
Yeah. So the diagnostic armamentarium to give the right diagnosis, number one, right? And
Hmm.
then number two, the right information around that diagnosis. And then, as you say, number three is kind of back to another theme of our time together, and that is to really make sure you get out of the lab and participate in leadership and thought leadership for the health system that you serve. And it's having someone at the other end that can to your point, receive this information and know what to do with it. It's also thinking about participating in formulary discussions, right? To make sure that the right other antibiotics and antimicrobials are available if resistance is detected. And then also even thinking about infection control in your hospital, because it's also really important that these patients with um, resistant bugs get you follow proper isolation protocols if appropriate, so that you don't have spread of a multi-drug resistant pathogen within the hospital with nosocomial infections. So I mean, it really
Right.
spans the gamut.
It really does. And one thing we didn't even talk about is making sure you have the tests that detect the resistance mechanisms that is appropriate for your patient population, making sure that your MIC breakpoints are up to date if you're looking at specific antibacterial agents, and also making sure that you're testing for the antibiotics that are being used. If there are new antibiotics, can you provide the information on whether that patient's infective in cause of their infection is susceptible to that new antibiotic, and that could be done locally, or it could be sending it out to a larger reference lab if it's a really new antibiotic. But those are all things that we as laboratory leaders need to think about. Are we
That's
providing appropriate care for our patients?
that's right, and connecting that into the patient care environment. So going all the way back even to your point about some of these tests are just not going to be sustainable in some healthcare settings because of the
Right.
volumes and things. So if you choose a send out lab, a reference lab to send those to really be asking information around the completeness of their antimicrobial testing. Also, turnaround time, to your point, becomes really important as well. Understanding how quickly you'll get the results. So particularly if you're in a setting where the patient's in a high acuity care, where they might be receiving a broad spectrum antibiotic, which happens pretty frequently because of the concerns around antimicrobial resistance. The more rapidly you can get someone off of those broad spectrum, it's better for the patient. It's also better, again, to prevent resistance. So there's all kinds of things to think about, and because we are sort of at the center of the decision making, both for the detection of antimicrobial resistance and establishing the systems that we can respond as quickly and as effectively as possible.
Yes, so true. And I like what you said earlier, Bill, that there there are advances that are continuously made. We have advances to further support early pathogen detection. We have new rapid multiplex tests where you can get an answer in an hour or even less for certain resistance genes. We have rapid tests that can be used in a point of care setting to tell if someone has a viral infection versus bacterial. We even now have metagenomics, which could look at even human host response to see if it looks like a viral infection or not. And then, as you mentioned, AI solutions. I think artificial intelligence does offer some promising ways to scale diagnostic solutions globally. So that should help combat this global issue more effectively.
Yeah, let's hope so. And I know that there's a lot of your colleagues in the clinical microbiology division that are thought leaders in this, Dr. Patel and others, and
Yes.
microbial stewardship. I mean, that it all also boils down to that. For those of the listeners that are in a position where they could contribute to that, I think it's really important because they tend to be a lot of this stems from overuse of or certain antibiotics and antimicrobials.
Yes. We're part of the whole system, a critical part.
That's right.
Well, Bill, it's always great talking to you. Thanks for the great discussion, as always.
Yeah, yeah. Thank you for the great discussion. So I learned something every time. It's great.
Same here. Until next time, take care.
You too.
Welcome to our first deep dive on Answers from the Lab. I'm thrilled to be joined today by my colleague, Dr. Paul Jannetto, who will share insights into how diagnostics are shaping the future of therapeutics. Dr. Jannetto is a professor and consultant with me in the Department of Laboratory Medicine and Pathology at Mayo Clinic. One of the areas he focuses on that I find fascinating is therapeutic drug monitoring. Thank you for joining me today, Dr. Jannetto.
Thank you, Dr. Pritt. It's a pleasure to be here.
So, yeah, let's start with a question about your journey. What sparked your interest in therapeutic drug monitoring?
Well, my journey actually began back when I was in high school.
Oh,
I had
wow.
been working at a small mom and pop retail pharmacy called Dietrich's Drugs for 6 years. And I did everything from counting out dispensing medications to stocking shelves. And back then, we even rented out VHS videos. Um,
Wow.
And, and so I actually ended up developing in that job relationships with customers who were taking numerous medications and constantly struggling to find either the right dose or the right medication that provided them the therapeutic benefits without significant side effects from those medications. And at the time, the pharmacist actually talked me out of going to pharmacy school. But that's where I really sort of decided upon a career in laboratory medicine where I could focus on therapeutic drug monitoring. And I really felt that was a good fit for me.
Thanks, Dr. Janetta. That's a great story. Can you explain for our listeners what role therapeutic drug monitoring has in delivering better therapeutic solutions?
Absolutely. So therapeutics really begins to dive in what's sometimes called precision or personalized medicine. And most medical treatments are designed for the average patient, sort of a one size fits all approach. And that may be successful for some patients, but not for all. And so precision medicine is this new, more innovative approach to tailoring both disease prevention and treatment that takes into account differences in people's genes, their environment, and lifestyles. And the goal of precision medicine is to target the right treatment to the right patients at the right time. So with therapeutics, this generally involves both pharmacogenomic testing and therapeutic drug monitoring or management. And pharmacogenomic really kind of combines the pharmacology and genetics to reveal how individual genetic variants or mutations may affect a person's response to a drug. This testing is really critical because it can help predict or sometimes even explain the effectiveness and safety of medications while minimizing adverse drug reactions and optimizing those therapeutic beneficial outcomes. So pharmacogenetics testing can help physicians select the best medication, or at least the starting dosage, to really get to that optimal therapeutic levels and therefore optimal therapeutic outcomes. In contrast, therapeutic drug monitoring or management is really the practice of measuring either the parent drug and or the metabolites of that drug to ensure the appropriate concentrations of that medication are achieved to get the desired clinical outcomes. And it provides really an objective measure of adherence or compliance, as well as the assessment of therapeutic effectiveness. And so if you integrate TDM serially into the treatment workflow, clinicians can now measure the drug concentrations at designated intervals to help ensure that accurate or the target drug levels are reached. And this is all because it's based on the fact that there's a relationship between the dose that's given and that serum or blood drug concentration, and that drug concentration correlates with either efficacy or toxicity. And so TDM traditionally begins after you start the medication, and several factors have to be considered when you're interpreting the results, such as the sampling time as in relationship to when you took the medication, the dosage, the patient's response, any potential drug-drug interactions or other medications they're on, and even the desired therapeutic range for that patient. But the goal of TDM and pharmacogenomics is really to optimize clinical outcomes in patients.
Well, thanks, Dr. Janetta. That's a great description, and those are good definitions for us to keep in mind. And I'm sure that things have changed since way back when you were in the pharmacy working on stocking shelves and selling VHS tapes and giving people advice. And so, looking back on your career, how have advances in therapeutic drug monitoring and pharmacogenomics benefited patient care?
Yeah, a big thing for me in therapeutic drugs has really been the use of mass spectrometry to be able to develop sort of laboratory developed tests has really helped advance the field of TDM because previously we really only had a limited number of FDA-approved, typically immunoassays, for a limited number of medications. And the problem with some of those assays is that the antibodies used had various lack of specificity. They would cross-react with a lot of the inactive metabolites or other drugs. And so they weren't really giving us accurate results. And we were truly reliant on diagnostic companies at that time to bring up and validate assays and take them to the FDA. And now, with using mass spectrometry-based techniques and laboratory-developed tests, we really can validate assays for the latest or newest class of medications that are approved for our patients. And that offers superior sensitivity, so we can measure down to lower concentrations, as well as that specificity that we didn't have with those immunoassays. And we don't have to wait for years for an assay to get FDA approved. We can actually, when the drug gets approved, have a companion diagnostic there to help monitor our patients. For me, I think one of the earliest examples of this was for a drug class that's known as the immunosuppressants. So this includes drugs like cyclosporin, tacrolimus, sirolimus, everolimus. These medications have a very narrow therapeutic window, and they're used in solid organ transplant patients. And if you have subtherapeutic concentrations of these immunosuppressants, this can cause a person to reject that brand new organ that they just got transplanted. Well, too high of a drug concentration can actually cause toxicity and damage that brand new organ that they just received. So as a result, these patients are on lifelong therapeutic drug monitoring for these medications, because as a patient ages, or your renal or liver functions change, right? Or maybe you start another medication, you could have potential drug-drug interactions. You may need to adjust that dose to maintain those therapeutic levels. The original assays that I started using back in the day were all immunoassays to measure these immunosuppressants. And again, for example, for cyclosporin, it had varying degrees of cross-reactivity with the over 40 different metabolites that people produce. And that led to actually about results that were about 40% higher than if you just were to measure parent cyclosporin alone. And that made it really difficult to manage patients. Now, with our LC10 and mass spec-based assays, where we have that specificity and sensitivity, we can reliably only measure the active parent compound, in this case cyclosporin, and better manage our patients.
Wow, that's really impressive. And I love how you're bringing it back to the patient, why it's so important for us to have that accurate result and then to be able to offer these tests and not have to wait years and years when a new drug becomes available. So, as this patient care is evolving, how has the role of laboratorians changed, if at all?
Yeah, so that's an excellent question, because I really think that laboratorians now really have to work much closer with our clinical colleagues and teams, and we really have to integrate ourselves and become an active member of the healthcare team. In fact, we get to partner with our clinical colleagues in medicine and pharmacy, and that enables me, for example, to develop the next generation of laboratory tests to help advance personalized medicine. I'll give you an example. So sepsis affects about 1.7 million Americans annually, and unfortunately it has about a 25% 30-day mortality rate, so not very good. And beta-lactam antibiotics, things like meropenem, cefepime, piperacillin, tazobactam, these are first-line treatments for patients with sepsis due to their broad efficacy and safety. And we use them in about 70% of all our critically ill patients, even here at Mayo Clinic. Well, beta-lactams can be life-saving. Unnecessary, imprecise, or inappropriate beta-lactam usage can contribute to antibiotic resistance. In fact, we see that standard dosing often results into over 30-fold differences in drug levels between septic patients. And as you know, beta-lactam antibiotics exhibit time-dependent killing. So as a result, the therapeutic targets that we're trying to achieve are based on the percent of the time that that concentration of that drug exceeds that MIC, or the minimum inhibitory concentration of the organism, during that dosing interval. So we want 100% of the time that drug to be above that MIC concentration, right? So we can kill that organism, right, and get the patient better. Well, I partnered with a colleague here at Mayo Clinic, Erin Barretto from pharmacy, and with her leadership and numerous other clinical collaborators here at Mayo Clinic, we actually developed and validated a beta-lactam LC10 and mass spec assay, because there wasn't a commercially available assay on the market. And once we had that assay, we actually did a pilot TDM study here at Mayo Clinic on both adult and pediatric patients in our ICU who are taking a beta-lactam antibiotic. Now, initially, our patients are dosed or treated empirically, right, until culture and sensitivity results come back to give us that MIC target that we need to hit. And in this study, we used our new assay to measure both peak and trough beta-lactam antibiotic concentrations. What we found was that approximately 73% of adults did achieve therapeutic concentrations. However, that's not 100% of our patients, right?
Right.
And so of those who didn't achieve target levels, 20% of those actually had too high of drug concentrations. They kind of exceeded some of our safety thresholds. So we had to de-escalate or de-intensify the dosage and lower the dose. While the other portion of patients were underdosed, so they weren't getting high enough concentrations to be above that MIC. In total, even though we had about 73% that were in that therapeutic range, they ended up tweaking or adjusting the dosages of about 40% of all the adults in that study based on our TDM assay that we had for beta-lactams. In our pediatric population, here we only achieved therapeutic levels in about 56% of our patients. So of those who we didn't achieve the therapeutic level in, 36% were too high or exceeded that safety threshold, and the rest were actually being underdosed. So we were actually underdosing a significant portion of our pediatric patients. And again, there's risk of not treating the infection, right? Antibiotic resistance developing. So in total in that cohort, about 59% of the pediatric patients ended up actually having their dosage tweaked. And then on follow-up TDM, when we would repeat it after that new dose adjustment, we actually saw that we got from that initial 56 up to over 80% of the patients being in the therapeutic range. So it was a good example of showing how when we have a test or a tool like TDM to do beta-lactam drug concentrations, we could get our patients to a target and have that better outcome.
That is an amazing story. And it really highlights how you, in your role, you're working directly for patient care. You're working with the pharmacy. We in microbiology are also interested in sepsis and antibiotic use. So we talk about the drug-bug combination. So we find out what the bug is, the bacterium. Our colleagues in infectious disease give the drug with our guidance. And then you're helping them monitor the drug to make sure it's appropriate. And obviously too low isn't enough. Too much risks resistance. So we're all working together for the patient. And that's one thing I always tell people that's so great about our jobs is it's all about patient care. We're not just hanging out in the lab doing research. It's all about the patient.
No, I
So
think that's
yeah,
absolutely right. Because, in the end, it's all about collaboration, right?
yeah,
As you mentioned, between a whole bunch of different players. And the lab is one important piece of that healthcare member team with all our cohorts. And really, it's that collaboration that now, in the case of beta-lactams, TDM is now standard of care here at Mayo for beta-lactams. And we're actually improving target attainment in various populations with high pharmacokinetic variability. And this is ultimately improving patient safety and effectiveness of our therapies. So
yeah, that's fantastic. Well, looking ahead, what's the next frontier? What innovation should our listeners keep an eye out for, in your opinion, over the next, say, 2 to 5 years?
Well, as you know, the lab generates a ton of patient data.
Right.
And that includes our pharmacogenomic data and even our therapeutic drug concentrations and data, right? And so what we're already starting to do is partner with our clinical colleagues again and looking at how do we use that data. And in the case of TDM, how do we do population pharmacokinetic modeling? And so again, working with my colleagues from pharmacy and the clinical practice, Aaron Barretto has actually developed using our data an upfront cefepime dosing algorithm that in simulation, when we do simulations, actually prevented subtherapeutic levels in the first 24 hours of sepsis without increasing toxicity risks. And we're able to incorporate other laboratory data, including things like serum creatinine and cysteine EGFR, cystatin C EGFR categories, weight-based adjustments, flexible dosing options such as loading doses and extended infusions. And so we can take all of this laboratory data and create these different algorithms that ultimately have improved pharmacokinetic target attainment, so we can actually reach therapeutic levels while reducing those levels of toxicity. And so what we're doing now is looking to prospectively validate some of these algorithms that we're generating in our real-world patient practice.
I love how this is all real world practice for personalized care. We're talking about individual patients. Maybe someone has less absorption. Maybe they're on other drugs and there's drug-drug interactions, as you mentioned. It takes it to that whole level of personalized healthcare using those types of tools. So very fascinating stuff.
An exciting time.
Yes, exciting times. Well, thank you again, Dr. Jannetto, for joining us again. We love having you here, and I'm sure we will have lots of things to talk about in the future.
Thank you very much.
Let's wrap up with the top takeaways and how to learn even more on the topics we've discussed. In our news, we were excited to finally share our refreshed approach to answers from the lab, and we shared the latest news from the World Health Organization on antibiotic resistance, highlighting the importance of fast and accurate tests in directing effective therapy. Then in our deep dive, Dr. Paul Jannetto took the role of diagnostics in effective therapy a step further by discussing the growing role of pharmacogenomic testing in personalized medicine. My takeaway was how this is really expanding the role of laboratorians in patient care. And if you want to learn more about therapeutic testing at Mayo Clinic, we'll include helpful links in the show notes. Thank you for joining us today. I hope you find this new format valuable and that you will join us for our next episode when Dr. Bill Morice will join me for both the news and a deep dive on creating successful collaborations. You've been listening to Answers from the Lab. We're glad you joined us. If you haven't already, hit subscribe so you never miss an episode. And if there's a topic you'd like us to explore, email Mayo Clinic Labs at mayo.edu. And while you wait for the next episode, connect with us on social media or visit mayocliniclabs.com for more insights, discussions, and resources.